welcome back, and today we will be doing DNA synthesis. But in essence, I honestly think snacks have got to be part of my DNA, because how do I live without it? So I am gonna explain an analogy that makes it easy for you to understand how DNA synthesis works. Then we're gonna switch it up and only use the biology terminology to ensure that we're always getting our marks in any given exam. So to start off with, let's say we've decided to have our snacks, peanuts and raisins. So peanuts would be our hard shell. So let us do 1, 2, 3, 4, 5, 6 peanuts. And these correspond with, let's do an orange, with 6 raisins. 1, 2, 3, 4, 5, 6. All right. So for me to be able to synthesize, to make more of DNA, let me just do the analogy. So let's say we have a bowl of peanuts and raisins, and I want to, from one bowl, I want to now make 2 bowls, ensuring that we have the same amount of peanuts and raisins. What I would do is that I would put all the raisins, 2, 3, 4, 5, 6, in one bowl. Then I'd put all the peanuts in another bowl, so that if I got a new packet of peanuts and raisins, from the packet for the first one, I would only take 6 peanuts, and from the packet I would only take 6 raisins. So that my final product then is 6. So even though my packet has tens and thousands of peanuts and raisins, I will not know how to do the exact same bowl if I do not have what we call the word template of the original bowl. So follow with me. My original bowl gave me the amount of peanuts and raisins. For me to be able to make more of them, I need to have that template, that baseline, the minimum required in each bowl. I only need the 1/2, because from the 1/2, all I do is match it up with its like relative and partner, and then I have the exact same amount and the exact same codes, because what I've done is synthesized or copied. Okay, so we basically just copied. So now let's redo this, but now I'm going to put in some biology terminology. I'm going to use the same one and then I'll rewrite it just in bio. So my first bowl, right, my initial bowl, so this would be found, let's make it in red because I'm just going to mess up my picture now. So this is obviously found now in the nucleus, right? Everything would be found in the nucleus, but let's zone in on the red, right? In this, I'm going to unzip. So when I unzip, I open up my DNA, which was a double helix, into strands. One strand is one ball and one strand is another ball. On the one strand, I will have a template, right? So this template was my raisins. On the other strand, it forms the template. So on this template, it was my peanuts, right? Then I have what we call free nucleotides. So my nucleotides are free floating. So it's the packet that has like many possible nucleotides that only bind to its nucleotide pair. So it'll only bind to its pair. Meaning that the 6 raisins will only bind with 6 peanuts. The 6 peanuts will only bind with 6 raisins, right? So therefore, we know that peanut A, adenine, will bind with raisin T, thymine. Raisin C, cytosine, will bind with peanut G, guanine. So we know the A will bind with the T. So now we know that A will bind with the T. Or we know that C will bind with the G. Does that make sense? This would obviously then be a complete copy and paste of the original DNA strand. All right. Then the two strands zip up again. So now we get our hydrogen bonds again formulating. So we'll get our hydrogen bonds. And then now we have two identical bowls of peanuts and raisins, but it's also two identical DNA strands. All right. So let's redo this with a whole lot of biology. So let me go back to blank. So initially we have our double helix strand. I'm going to choose about 9 letters just so we can follow along. Just want to do the first side first. 1, 2, 3, 1, 2. I think I can put another one. Okay, let's do this so we follow along. So we have 1, 2, 3, 4, 5, 6, 7, 8. Let's do 9 there. Okay. So on my first side, let's do it all blue. So let's say this is an A, this is a C, this is an A, this is a, I'm saying a C and I'm writing a G. This is a G, this is a G, this is a C, this is an A. Oh, I'm saying the letter, but I'm actually writing its base there. T and A. All right. So this is what we have on our first strand, right? Then we obviously know that the strand would be binded on the other side to its opposite, or its, let's not say opposite, so it's base pair. So let's do all our pairs on the other side. So you can pause right now and you can guess the other side with us so that it would be good practice for you. All right, so the A will bind with our T. Our G would bind with our C. You see, I think one thing and I write the other thing. Our G will be bound with our C, our A with our T. Good. Our G would be, so obviously it's a helix, so just follow with me. It's still wind it up. Our G would bind with our C. Our G would bind with our C. Our C would bind with our G. Our T would bind with our A. Our T would bind with our A. And our A would bind with our T. So this would be our double helix strand before DNA synthesis begin. Our first step, obviously, we're going to have is that it will unwind. So now I'm going to do one strand there. And I'm going to do one strand there. So then we understand 1, 2, 3, 4, 5, 6, 7, 8, 9. Our first step is that it's going to unwind. So each DNA strand, remember it was a double helix strand. Now each strand will form a template. So it will become our 1, 2, 3, 4, 5, 6, 7, 8, 9. What we require to then make our, so let's just copy. So this is A, this is G, this is A, this is G, this is G, this is C, this is, sorry. Y'all know that helix is helixing. Just want to go back to it. This is T, this is T, this is A. It's just the colors helped actually. This is T, this is C, this is T, this is C, this is C, this is G, this is A, this is A, this is T. So we have a double helix strand. Our double helix strand will unwind, right? As it unwinds, we then now have two strands that we are going to use. We then have free nucleotides, right? So these are anything that is able to now bind with what we have. So let's just put it back in red so that we can just follow along. So we have an unwind, we have a template, we have free nucleotides, just so you're following with me. All right, so that's what we have. Let's just remove that just so you're with me. Okay, so let's go back into black. So now our free nucleotides, our free nucleotides will basically, T will bind with A, C will bind with G, T will bind with A, C will bind with G, C will bind with G, G will bind with C, A will bind with T, A will bind with T, T will bind with A. Same thing on the other side. A will bind with T, G will bind with C, A binds with T, G is C, G is C, C is G, T is A, T is A, and A is T. Now, if you look at it, the first strand, let's choose another color so that you can see what I'm talking about. The first strand there created now a new DNA, and if you look at it, it has the exact same nucleotides in the exact same sequence as our initial DNA. Additionally, our second DNA strand looks exactly like our original DNA. So does that make sense? So we use our initial DNA. Each strand becomes a template for free nucleotides to bind to it. This then creates the new DNA. So basically a new DNA, but it's exactly an identical copy of the initial DNA. This will then zip up again. So now we'll have a zip up. So that means the bonds come back together again. It will wind up again like double helix, and then there we have two now DNA strands instead of that one initial one that we had. However, sometimes we want to be able not just to increase the DNA molecules within, but now we need the DNA molecule out of the nucleus. And so you must understand, if we are going out of the nucleus, the same way we were able to do 2 within the nucleus, we are only going to need 1 strand to go out the nucleus. So we're going to do the exact same thing. Let's just rewrite it again. So we're going to go back into black. So we again will have our double helix strand. This time I'll only do 4 because this one can get okay, let's do 6. This one can get a little bit more weird if we don't follow along. So it's 1, 2, 3. There's 1, 2, 3 there. So here I'm going to do a G, an A, a T, an A, and a C. We understand that that would be binded to that will be binded to a C, a T, and a T, a T, and a G. Now the first step again, like we did, we need to unwind the strand. So let's take one strand. Let's take the black strand because that's better for when we start to switch it up a bit later. So then we're going to take one strand. So my strand from the DNA was GA, T, A, C. I haven't changed anything. All I did was unwind my DNA. So I just unwind the DNA. Then I'm going to use my template strand. Right, so this is my template strand. I'm going to need free nucleotides. So these are the free nucleotides. So let's do the free nucleotides in another color. Let's do a nice bright green. My free nucleotides will then be my mRNA. Okay, so my free nucleotide is actually an RNA strand. So now let's first make it easy. So first my free nucleotide. So I'm just going to do a C, a T, an A, a T, a T, a G. However, because this is an RNA strand, you must remember that now we do not have thymine anymore. We have uracil. So that means every single time you see a T, this would have actually been A binded with a U, A binded with a U, A binded with a U. So that just takes the place of our T, right? So now we have our mRNA. But another way to think of mRNA, it's our moving mRNA, like it's the moving RNA strand, all right? So the mRNA strand will then leave my nucleus through my nucleo. It'll leave the nucleus. Sorry, I'm just trying to draw a nice little, leave the nucleus into the cytoplasm, where transcription would then have ended. Oh, sorry. Transcription would then have ended. So that's all that is. So the first one is the copy, right? So let's repeat it again. We have a double helix strand within the nucleus of the cell. We have to unwind it because we need to open it up so that we can copy. You can see right now I'm getting confused when it's like unwinded, even though it's so nicely color coordinated. So let's unwind it. Use that free template strand, bind it to free nucleotides on an mRNA strand. That should then trigger, again, the differences between RNA and mRNA, which is only that one nucleotide that is not found in RNA. You can just copy and paste it the same way we've done it, and then flag all your T's and change them to U's, or just remember and like correctly change. Then the mRNA strand leaves the nucleus. Done. That is the first part done. Translation, as you can imagine, is us now doing or translating, doing exactly what we said we're going to do, which is to make our amino acids. Now our mRNA strand sits in our cytoplasm. I should have did it in the same format because we've been doing vertical just so it doesn't look different. So let's keep it vertical. So now we're going to go 1, 2, 3, 1, 2, 3. So now we have a C, a U, and a U, a U, and a G. All right. So basically now all we're going to do is, again, we are going to bind. However, this one is not in essence free nucleotides. All right. So now when we're in the cytoplasm, what floats around is pairs of 3, or not pairs, but a triplet organization. So that would now be our tRNA. Okay, so it will come in a pair of 3 to give you one coded amino acid. It will come in a pair of 3 to code one amino acid. So on that code, you will see a C, an A, and a U, an A, an A, and a C. However, it is not what's on or what's been transcribed that's important, but really the transcription is that coding activates an amino acid. So basically what happens is one amino acid would start at 1, right, at that C. It'll give one amino acid. Let's say that amino acid is pink. Then it moves down to the next three. That amino acid is blue. Then it moves down to the next three. That amino acid is brown. So by the time it's done reading the whole mRNA strand, the amino acid has coded pink, blue, brown, green, orange. Pink, the next mRNA strand will be a pink, green, blue. So all we're saying is that now it's taken three codons, anticodons. We don't want to confuse you as yet. Get the big thing going. Then I'll repeat a little bit. So it has taken everything from our mRNA, coded it to give you one code, and that one code is coming through our tRNA to give you only one piece of our amino acid chain. All right, so again, I just popped in that word of codons and anticodons. So another way to remember it is the code would be in our tRNA, because as you can see, it's like held within something. So then our tRNA is our code, because that is it's our final thing. So it's our codon, and our mRNA would have been our anticodon, because as you can imagine, it's opposite our original copy. So now, just in summary, so that we kind of are together, just remember that I think in this section, the biggest thing to remember is the differences between the RNAs, because we have different types of RNAs. Remembering as well the main differences between DNA and RNA, to remember that nucleotide. But also remembering that we have to unwind for nucleotides to bond. Or like in the cytoplasm, we don't unwind anymore. So then all the tRNAs, all the codons move around the cytoplasm, and all they do is bind to another, not bind, but they read. So this actually moves down. Okay, so it's not like, it's one that codes and then like transcribes. So it reads through the strand. But the biggest thing obviously is just to understand it and see how it goes. So it's very nice to see how DNA synthesis occurs within the nucleus. And then as we move out of the nucleus, we are going to need an RNA strand. So we use our mRNA strand to move us out. And then we use our tRNA to help us code for an amino acid. So please sit with this a little bit and see if it makes sense. And if it does make sense, let us then jump onto another section, a little bit of application of how DNA would be replicated within a cell. Maybe something like meiosis. Let's see how that's